Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing Karnataka State Eligibility Test Life Science question paper, paper 2 conducted in the year 2018. So coming to the first question. An open reading frame contains 333 nucleotides. The number of amino acids in the protein synthesized from this open reading frame will be. So one codon is one of the 333 codons will be a termination codon. So the 333 minus 3, that is 330, because termination codon doesn't add an amino acid. So you have to subtract 3 from 333, you get 330 codons. Sorry, nucleotides. No, yeah. Then 3 nucleotides code for 1 amino acid. So 330 nucleotides will code for 110 amino acid. So the answer is option B. Then next one. Color blindness is an X-linked recessive disorder. If a child is born to a normal father and carrier mother, what will be the probability that the child is colorblind? So color blindness is X-linked recessive and the child is born to normal father and carrier mother. So the normal is capital C and the small c is the recessive phenotype. So this is the normal father. X capital C is Y is the normal father. So these are the gametes and these are the offspring that are born. So what will be the probability that the child is colorblind? So out of these, one is carrier and the other will be colorblind and the other two will be perfectly normal. So 1/4 of the children will be colorblind. So answer is option C, that is 1/4. Then next one. Plant movement take place in response to touch stimulus is called as. So it is thigmotropism. It's directional growth movement which occurs as a mechanosensory response to touch stimulus. So the answer here is option A, that is thigmotrophic movement. Then next one. Which techniques are used to study the transcription process in the cell? So transcription is synthesis of RNA. So northern blot and in situ hybridization can be used. So answer is option C. Northern blot is for detecting RNA molecules. Then next one. Radioactive sulfur was used to label the T4 phage coat protein. When the phage was infected to E. coli cells and the cells were precipitated by centrifugation, S35 labeled protein was found with. So protein coat will not enter the infected cell. This is P4 phage. So when the phage was infected to E. coli cells, so the phage was infected to bacterial cells. The protein coat doesn't enter the infected cells. So they will be found in the supernatant. Answer will be cell supernatant. Answer is option B. Then metagenomics deals with. So metagenomic, it is the study of the structure and function of the entire nucleotide sequences isolated and analyzed from all the organism. That is, if you are taking the case of microbes, it is the study of the structure and function of the entire nucleotide sequences isolated and analyzed from all the microbes in that population. And metagenomics is often used to study a specific community of microorganisms such as those residing on the human skin, in the soil or in water sample. So the answer here is culture independent analysis of biodiversity. So metagenomics deals with culture independent analysis of biodiversity. Next one. Bats belong to phylum. They belong to phylum Mammalia. Answer is option D. Which element is replaced by strontium 90 in bones? So strontium 90 chemically resembles calcium and it gets assimilated in the bones and teeth and causes radiation injury by damaging the bone marrow and impairing with the formation of new blood cells, and it can even lead to cancer. So strontium can be replaced with calcium can be replaced with strontium 90 in the bones. Then ergosterol is specific to. So ergosterol is a primary sterol in the cell membrane of filamentous fungi. So the answer is option D, that is filamentous fungi. Then identify the right sequence of IgG gene sequence joining that result in the production of functional heavy chain of immunoglobulin. So heavy chain of immunoglobulin, it is coded by VDJC sequences. Answer is option D. Then almost all major animal body plants seen today are found in which of the earliest fossil? So in the Cambrian period, there was an explosion of different types of living things. Most of the major phylum of invertebrates suddenly appear in the fossil record. But there are no transitional fossil for these phylums which are found in the Precambrian rocks. So Cambrian period almost all major animal body plants seen today are found in the earliest fossil of the Cambrian period of the Paleozoic era. Answer is option A. Then trisomy of chromosome 18 in human is. It results in Edwards syndrome. Answer is option D. During abiotic stress, plants accumulate. During abiotic stress, plants accumulate proline, because proline acts as an osmoprotectant and also a ROS quencher. Then which of the following is not a living fossil? So Archaeopteryx is not a living fossil. It is a connecting link between birds and the reptiles. So answer is option D. Then next one. Digger wasp will immobilize a prey, drag it to the hole in the ground, enter the hole, inspect it, place the prey in the hole and lay an egg on the prey and close the hole. If during this cycle of events any one of the actions gets interrupted, it will go through the entire sequence once again. This is an example of. So first option they have given as innate behavior. Now what is innate behavior? It is genetically hardwired in the organism and can be performed in response to a cue without any prior experience. And imprinting is what you see in certain animals, that is ducks and geese. When the duckling hatches, they imprint on the first adult animal they see, and typically that is their mother. Then learned behavior is one that an organism develops as a result of experience. And trial and error is problem solving method in which multiple attempts are made to reach the solution. And trial and error, it is a trying method, that is observing it for weeks, and if you don't, trying a new method. So the answer here is option A, that is innate behavior. This is an example of innate behavior, and this experiment, it was conducted by French naturalist J. Henry Fabre, which illustrates the character of innate behavior. The next one. During bacterial conjugation, the directional transfer of DNA from donor to recipient occurs when F factor is integrated into donor chromosome. So answer is option B. The directional transfer of DNA from donor to recipient occurs when F factor is integrated into chromosome. Answer B. Then flow cytometry generally used. It is used for identification of cell types. Answer is option D. Then which one of these animal groups have the following characteristic? A body with trunk and tail and notochord extending from the rostrum to the tail. So this is seen in the case of Cephalochordata. Answer is option A. Then next one. Which of the following cells do not contain nuclei? So platelet or thrombocytes are small colorless cell fragments in our blood that form clots and stop or prevent clotting. And these cells, they do not contain nuclei. So answer is option B. The naturally occurring phosphoprotein is. So phosphoproteins are conjugate proteins and they are post-translationally modified by the attachment of phosphate groups, and naturally occurring phosphoprotein are milk protein casein and ovovitelline of egg yolk. So the answer here is option C, that is casein. Then which of the following is not component of MAP kinase pathway? MAP kinases are grouped into three families, that is the mammals they are ERKs, that is extracellular signal regulated kinases. Then JNKs, that is JUN amino terminal kinases, and P38 SAPKs, that is stress activated protein kinases. So what is left out is this one. Option A is MAP kinase. And option C is the answer, that is MEK. It is not a component of the MAP kinase pathway. Then transducin is a G protein involved in which of the following biological processes? It is involved in phototransduction, that is vision. Answer is option C. Then which of the following model organism gene mapping can be made by tetrad analysis? So tetrad analysis is well studied in Neurospora crassa. Answer is option D. Then ions can move from one cell to another directly passing through. So the gap junctions, these are channels that can pass ions and small molecules from the cytoplasm of one cell into the cytoplasm of an other cell. So answer is option B, that is gap junctions. Then which of the following is an example of genomic variation? So copy number variation are important component of genomic diversity as single nucleotide polymorphism. So copy number variation is a SNP. Answer is option A. Then animal cell culture is quite popular in production of. So it is quite popular in the production of vaccines. Answer is option D. Then Beadle and Tatum showed that each kind of mutant bread mold lacked a specific enzyme. These experiments demonstrate that. Genes carry information for making proteins. Answer is option A. Then next one. In bacteria, if bacteria doubles in 5 minutes, what would be the number of bacteria at the end of 25 minutes if you start with 50 bacteria? So final number of cell is equal to 2 raised to n into initial number of cell. N is number of generation. So here it doubles every 5 minutes. So 5, 10, 15, 20, 25. So number of generations will be 5. So 2 raised to n is equal to 25. 2 raised to 5, that is equal to 32. So 32 into initial number of cells, that is 50. That will give you 1600. Answer is option D. Then next one. During which stage of development an embryo becomes tryptoblastic? So it is during the gastrulation stage that the embryo becomes tryptoblastic. Answer is option C. Then ecological footprint of a land is an estimation of. It is an estimation of area of land per capita to meet actual demand on resources. Answer is option C. Then unlimited population growth is often prevented when death rate increases as population density increases. It is an example of. So if you see a positive feedback loop, there is an increase in the population lead to more number of birth, which increases the population even more. That is in the case of positive feedback loop. With the negative feedback loop, an increase in the population reduces the food supply, and less food means more death and fewer birth. So this is an example of negative feedback. Answer is option A. So unlimited population growth is often prevented when death rate increases as population density increases. Then which of the following bacteria perform mixed acid fermentation? So mixed acid fermentation, it is a biological process whereby the 6-carbon sugar, that is glucose, is converted into a complex and variable mixture of acids. and such as lactate, acetate, succinate, fumarate, formate, ethanol, gaseous hydrogen and carbon dioxide. And mixed acid fermentation, it is performed by Escherichia coli. Answer is option C. Then hydroxylysine and hydroxyproline in collagen are the result of post-translational modification of lysine and proline. That's correct. Answer is option A. Then next one. A substance that mimics the cellular effect on a natural compound is known as, it is called as an agonist. Answer is option B. Then lichens are combination of algae and fungi. Answer is option A. What would be the most likely conformation of the following peptide? So it is having glycine, proline, methionine, leucine, aspartic acid, phenylalanine, proline, lysine, alanine. So it will be mostly a random coil. So answer is option D. Then obligate anaerobe means. Obligate anaerobe means they do not use oxygen as final electron acceptor. Answer is option B. Then next one. The plant viruses that multiply within their insert vector are called. They are called as propagative. Sorry, it is not insert vector, it is insect vector. Plant viruses that multiply within their insect vector are called as propagative. Answer is option C. Then neuron response after an immediate shock is an example of. So first option is paracrine signaling. So paracrine signaling is a form of cell signaling in which a cell produces signal that induce changes to nearby cells. And if it is signaling molecules, that is example is hormones, they are secreted by specialized endocrine cells and carried through the circulation to act at distant target cells, then it is called as endocrine signaling. And direct touch signaling, that is direct contact signaling, it is also called as juxtacrine signaling and involves communication between cells that are in direct contact with each other. So neuron response after an immediate shock is an example of synaptic signaling. Answer is option C. Then which of the following functions is not concerned with chorioallantois of mammals? So it is chorioallantois is placenta in mammals. And it is not concerned with locomotion. It is concerned with respiration, excretion, nutrition, but not with locomotion. Then late blight of potato is caused by, it is caused by Phytophthora infestans. Answer is option A. Then next one, which of the what would be the phenotype of Drosophila when the X chromosomal and autosomal set ratio is 0.67? So if the ratio is 0.66 or 0.67, it is called as an intersex. Answer is option B. If it is 0.5, then it is normal male, and if it is 0.33, then it is a meta male. So the answer here is option B, that is intersex. Then next one, the classical four-winged mutant fly Drosophila melanogaster resulted from. So mutation in the ultrabithorax gene causes homeotic transformation of the normally two-winged Drosophila into a four-winged mutant fly. So the answer here is homozygous for 3 mutant alleles of the ultrabithorax gene. Answer is option B. Then next one. Kogler and Milstein are associated with, so they are associated with hybridoma technologies for production of monoclonal antibodies. Answer is option D. Then high yielding and photosynthetically efficient plants are usually, and they are usually polyploids. Answer is B. Which of the following method can be used to enumerate the deer population in a forest? So it is line transect method. Answer is option B. Then yeast is an anaerobe as well as an aerobe. Answer is option C. Then Frankenstein food are products. So they are products from GMOs, that is genetically modified organisms. Answer is option A. The next one, a dense bacterial population caught in a tangled web of fibers sticking to a surface describes. It describes a biofilm. So biofilm are slimy layer of microorganisms that stick to wet surfaces. The next one, which of the following processes occurs exclusively in the cytosol of an eukaryotic cell? So glycolysis TCA. TCA occurs in mitochondria. Glycolysis fatty acid biosynthesis. That occurs in the cytosol. Fatty acid biosynthesis occur in cytosol. Beta oxidation in I think it is in mitochondria. And the TCA cycle and beta oxidation they take place in mitochondria. Then what is Bohr effect? That is the next question. So at a low pH concentration and high carbon dioxide concentration in peripheral tissues, the affinity of hemoglobin for the oxygen decreases and oxygen is released to the tissues. Similarly, in the capillaries, or not similarly, conversely, in the capillaries of the lungs, there is, as carbon dioxide is excreted and the blood pH constantly rises, the affinity of hemoglobin for oxygen increases and the protein, that is the hemoglobin, it will bind more oxygen for transport to peripheral tissues. This effect of pH and carbon dioxide concentration on the binding and release of oxygen by hemoglobin, it is called as Bohr effect. So the answer here is effect of pH on oxygen binding to hemoglobin. That is correct answer is option A. Then sarcolemma is plasma membrane of, it is plasma membrane of muscle fiber cells. Answer is option C. Then cancer arising from epithelial cell is called, it is called as carcinoma. Sarcoma is for a cancer of the bone or muscles. Leukemia is of the blood cells or blood cancer. Adenoma is a benign tumor. So the cancer arising from epithelial cell is called as carcinoma. Then historically, which was the first genetically modified plant for antibiotic resistance and was produced in 1982? It was tobacco. It was the first genetically modified plant for antibiotic resistance. Then a gene was cloned in tetracycline resistant locus of PBR322 plasmid. The plasmid was then introduced to E. coli and grown. Which of the following statements are true? So here the plasmid is a gene was cloned in the tetracycline resistance locus of PBR322. So tetracycline gene is disrupted. The one that is the gene which will degrade the tetracycline present in the medium is been disrupted. The plasmid was then introduced to E. coli and grown. Which of the following statements are true? Now PBR322, it has got normal PBR322 vector, not the one with the insert. It is having a tetracycline resistance gene and an ampicillin resistance gene. Now what they have done? E. coli grown in medium containing ampicillin but not tetracycline. So one is correct because I already told you that tetracycline gene has become inactive and recombinants will not grow in medium containing tetracycline, but they will grow in medium containing ampicillin since ampicillin gene is intact. So one is correct. Then next one, E. coli will grow in medium containing tetracycline but not in ampicillin. That is not correct. E. coli will not grow in tetracycline containing medium. Then the colonies that grow on tetracycline containing medium have the inserted genes. The colonies that grow on tetracycline containing medium will not have the inserted gene. They will be non-recombinants. So 3 is also not correct. Then next one. The colonies that grow in ampicillin but not on tetracycline will have the inserted gene. That is correct. So 1 and 4 are correct. That is option C is correct. Then 15 microgram of amylase, molecular weight 150 kilodalton, acts on starch to produce maltose. It has maximum velocity. If at maximal velocity, the enzyme released 6.84 milligram of maltose of molecular weight 342 per minute. What is the turnover number? I have done it in the next photograph. Okay, so you have got a 15 microgram protein of molecular weight 150 kilodalton, and it produces a product of concentration 6.84 milligram per minute, and the molecular weight of the product is 342. Now they have asked for what is the turnover number. So it is Kcat is equal to Vmax by total enzyme concentration. So moles of amylase will be 15 microgram per 150 kilodalton. That is 15 into 10 raised to minus 6 grams divided by 150 into 10 raised to 3 grams. That will give you 10 raised to minus 10. Now Vmax is equal to moles of product formed per minute. So that is equal to 6.84 milligrams or 6.84 into 10 raised to minus 3 grams divided by molecular weight 342, that is 2 into 10 raised to minus 5, and that is the Vmax. So Kcat is equal to Vmax by total enzyme concentration. So Vmax by ET, that will give you 2 into 10 raised to 5 per minute. So answer is option A. Then next one. Ground feeding squirrels have one squirrel to stand as sentinel, that is guard, to give alarm call if any predator is sighted. This action would put the life of the sentinel at risk. Yet the squirrels do it. This is explained on the basis of. So the first option is altruistic behavior. So altruism, it refers to the behavior that benefits another individual at the cost of oneself. That is, if you take the example of sharing your lunch with your cousin sister. So it is an altruistic behavior because it helps someone who is hungry, but at the cost of you being hungry. So that is called as altruism. Then reciprocal altruism is an altruism that occurs between unrelated individuals. And there is a promise of repayment of the act. That is the case of reciprocal altruism. Then group selection, it is defined as selection in which traits evolve according to the fitness of the group. And then finally there is kin selection, and kin selection occurs when animal engages in self-sacrificial behavior that benefits the genetic fitness of its relative. So ground feeding squirrels have one squirrel to stand as guard. So the guard squirrel is sacrificing, that is self-sacrifice for the benefit of the whole community. So this what does the sentinel do? It gives alarm if any predator is sighted, and this action can put the life of the sentinel at risk. So the squirrel is self-sacrificing itself for the benefit or genetic fitness of its relatives. So it is an example of kin selection. So answer is option D. Then pollinating insects and flowering plants depend on each other. The evolution of these two is explained on the basis of, it is coevolution. Answer is option D. And coevolution occurs when two or more species reciprocally affect each other evolution through the process of natural selection. Then next one. In the DNA replication experiments by Okazaki, although he interpreted its result to support semi-discontinuous synthesis of DNA, there were objections to this interpretation. Which of the objections was valid, requiring further experimentation? So, DNA replication experiments conducted by Okazaki, and he interpreted that the replication is semi-discontinuous, but there was objection to this interpretation. Now, which of the objection was valid, requiring further experimentation to be done? So the answer here is option D. That is, uridine is incorporated in place of thymidine. Removal of uridine leads to strand breaks. So this was the objection. So answer is option D. Then which one of the following statement is incorrect to define mutation? So mutation may or may not produce discernible phenotype. That is correct. A mutation can induce damaging effects on normal gene sequence. Yes. Mutations are important players during evolution. That is also correct. Chemical mutagenesis can be used to produce transgenic animals. So that is an incorrect definition of mutation. For creation of transgenic animal, three principal methods are DNA microinjection, embryonic stem cell mediated gene transfer, and retrovirus mediated gene transfer. Not chemical mutagenesis is used to produce transgenic animals. Answer is option D. The maturation promoting factor of cell cycle is, so maturation promoting factor, it is a heterodimeric macromolecule composed of cyclin B and cyclin dependent enzyme that stimulates mitotic and cellular division phases of the cell cycle. So it is a cyclin dependent kinase complex. Answer is option C. Then next one. Which of the following phylum has metameric segmentation? So annelid, arthropod and chordata are metameric segments, are having metameric segmentation. So answer is option C, that is annelida. Then which of the following transgenic animal was first developed for producing alpha-1 antitrypsin in the milk? So alpha-1 antitrypsin, it is a protein produced in the liver that protect the body's tissues from being damaged by infection. So, and this alpha-1 antitrypsin AAT is a protein that protects lungs and the liver from damage, and this was developed in which transgenic animal, that is sheep. Then next one. Which of the following compounds form chitin, the exoskeleton of insects? So chitin is an acetylated polysaccharide composed of N-acetyl D-glucosamine groups linked by beta-1,4 linkages. So the answer is nitrogen containing polysaccharide. Answer is option B. Then RNA editing is post-transcriptional modification that includes addition, deletion or replacement of nucleotides in the mature RNA. Which of the following molecules play a role in mediating this process? So in RNA editing, it is a post-transcriptional modification and it can involve either addition or deletion or substitution of nucleotides in the mature RNA. Now, which molecule play a role in mediating this process? It is guide RNA. Answer is option D. Then, the three-dimensional structure of living cells or tissues can be viewed in. So, it can be viewed using a stereo microscope. Answer is option D. Then next one. Which of the following is not a mating system? Polyandry. Polyandry, it is a pattern of mating in which male animal has more than one male mate. Polygyny is in which a male animal has more than one female mate. And polygamy is a pattern of mating in which an animal have more than one mate. And the sexual selection, it is a mode of natural selection in which members of one biological sex choose mates of the other sex to mate with, and also they will compete with the members of the same sex for access to the members of the opposite sex. That is sexual selection. So which of the following is not a mating system? It is sexual selection. Answer is option D. The next one. Neem-based biopesticide azadirachtin does not directly kill pest but alters the life processing behavior in such a manner that the insect can no longer feed, breed, or undergo metamorphosis. Azadirachtin interferes with the metabolism of. So azadirachtin interferes with the molting of insect and has growth regulatory and stimulant effects. So, and ecdysone is a steroid hormone secreted by prothoracic gland that is in its active form stimulates metamorphosis and regulates molting in insects. So, azadirachtin will interfere with ecdysone. Answer is option B. The next one. Which one of the following methods is used to detect the distribution of specific mRNAs within a cell? So RNase protection assay is highly sensitive technique developed to detect and measure the abundance of specific mRNAs in a sample of total cellular RNA. And in situ hybridization, it is used to localize sequence of RNA or DNA in a biological sample. And northern blot, it is for detecting specific RNA molecules among a mixture of RNA. And site mapping is a process of creating site maps that index information of your site for increased usability and easier website planning. So now they have asked which one of the following method is used to detect the distribution of specific mRNA within a cell. Distribution is it will localize the sequence of RNA in the biological sample. So it is in situ hybridization. Answer is option B. Then next one. Trees may be damaged by animals which rub against them, wearing a strip of bark right around the tree trunk and exposing the xylem. The tree will then. Now the xylem is exposed due to this animal behavior, and this will interfere with the transport of water and nutrients from the root to the leaves. So what will happen? The tree will die out. So it will die quickly because leaves are deprived of food and water. Answer is option A. The next one. The mechanism of signal transduction by steroid hormone differs from amine and peptide hormone because now steroid hormones, they can easily diffuse through the cell membrane because they are hydrophobic in nature. And these hormones, they will bind to its receptor in the cytosol or on the nucleus, forming hormone receptor complex, and this complex then enters the nucleus and it will bind to the target DNA and it will create mRNA through transcription, and then this is translated to proteins within the cytoplasm. Now what will happen in the case of amine and peptide hormones? Now these hormones, they are unable to diffuse the lipid bilayer of the cell membrane and they must pass on their message to the receptor located at the surface of the cell. The receptor then initiates a signaling pathway within the cell. So the mechanism of signal transduction by steroid hormone differ from amine and peptide hormone because answer is option A, that is they bind to cytoplasmic or nuclear receptor and affect gene expression. Answer is option C. Then next one. Which one of the following feature is not associated with grasses? So it is cellular endosperm. So helobial endosperm is common to grasses and cellular endosperm is mostly seen in the case of dicotyledons. The next one. Apoptosis, a process of cell death in living organism occurs. It occurs naturally as a part of normal cellular development. Answer is option B. Then the major amphibolic pathway in almost all living organism is. So the citric acid cycle, it is an example of amphibolic pathway because it functions in both the degradative, that is it will help in the degradation of carbohydrate, proteins, fatty acid as well as it act in the biosynthesis processes also. So citric acid cycle pathway. Answer is option D, that is a amphibolic pathway in all living organisms. Then student t-test is used for the comparison of two sample means. Answer is option A. Then precursor ethylene biosynthesis is methionine. Answer is option A. Then next one. Lady cells secrete. They secrete androgens. Answer is option C. Then almost all terpenoids are made up of. So they are made up of isoprene units. Answer is option B. Then which one of the following is not an example of extrachromosomal inheritance? So yeast peptide. Extrachromosomal inheritance means the DNA is present in the cytoplasm and not in the nucleus. So it is the DNA present in chloroplast or mitochondria, and they do not follow the Mendelian pattern of inheritance, and these are known as extrachromosomal inheritance. So they are asking which one of the following is not an example of extrachromosomal inheritance. So yeast peptide, yes, it is an example. Drosophila bari, it is not an example of extrachromosomal inheritance. Then next one. Which of the following cloning vectors can be used to clone 3 kb and 300 kb DNA fragments? Choose the correct order and vectors. So if you are using plasmid, it will be around, insert size will be around less than 10 kb. For bacteriophages, it is 9 to 15 kb. Cosmid, it is 23 to 45 kb, and BAC, it is greater than or equal to 300 kb. And PAX, it is 100 to 300 kb, and YAX, it is 100 to 3000 kb. So they have asked which of the cloning vectors can be used to clone 3 kb and 300 kb DNA fragments. So it will be, answer is option D, that is plasmids and YACs. Answer is option D. Then, during maturation of B cell, the immunoglobulin G arrangement occurs due to. So the answer here is site-specific recombination. So in site-specific recombination, the DNA molecules are rearranged by breaking and rejoining the strand at specific points. And site-specific recombination involves two short DNA sequences which may be within the same molecule or in different molecules. And the functional genes that encode the IgG light and heavy chains are assembled by recombinational events at the DNA level. And these events and the parallel events involving T receptor genes are the only known site-specific DNA rearrangements in vertebrates. So here what is happening is, this is the V, J and C sequence. Then DNA rearrangement by recombinases, it joins one V segment to one J segment. So one of the V segment is joined to the one of the J segment, that is V2 is joined to J2. The others are left as such. Then transcription and RNA processing as well as joining of this J segment, V2 J2 segment to the constant C segment. It will result in V2 J2C, and it will undergo translation and produces the immunoglobulin. So this is the variable region, this one, and this is the constant region. It will give rise to the variable V2, and J will give rise to variable region, and C will give rise to the constant region. So this is the resulting protein. So maturation of B cell, the immunoglobulin gene rearrangements occurs due to site-specific recombination. So answer is option C. Then ABO blood group in man is an example of. It is multiple alleles and codominance. Answer is option B. Goodness of fit is carried out by chi-square and goodness of fit. So answer is option A. Then what is meant by the word world in discussing floral meristem? So flower, they consist of different organs which occur in concentric rings called as whorls. That is sepal, petal, stamen, carpels. So the answer here is option B. That is flower consists of 4 different types of organs which occur in concentric ring called as whorl. Answer is option B. Then match the following. Escherichia pestis. It is black plague. A4. Then Mycobacterium leprae is Hansen's disease or leprosy. Rubella virus is mumps and Morbilli virus is measles. So the answer here is option A. The next one. The following is absolutely essential for the functioning of an ecosystem. That is, producers should be there as well as decomposers has also to be there. So answer is option D. Then which of the following is the right sequence of spermatogenesis? So spermatogonia, spermatocytes, spermatids and spermatozoa. Answer is option D. Then next one. Plasmodesmata in plants are similar to which one of the following structures of the animal cell? So plasmodesmata, they are intercellular pores connecting adjacent plant cells, allowing membrane and cytoplasmic connection, and also for trafficking, communication and signaling in plant development and defense. This is the function of plasmodesmata. And gap junctions, they are specialized intercellular connection between animal cells. And they directly connect the cytoplasm of two cells with and allow various molecules, ions and electrical impulses to directly pass through a regulated gate between the cell. So plasmodesmata in plants are similar to gap junctions in animal cells. So answer is option B. Then next one. Prokaryotic organisms have been divided into 2 domains, bacteria and archaea. The division is based on. So it is difference in cell wall composition, yes. Differences in cell membrane composition, no. Presence or absence of introns, yes. And presence and absence of amino acids, no. So the answer here is 1 and 3 are correct. So answer is option A. Then next one, match the following. Protozoa is cell wall is absent in case of protozoa. So A5. Eubacteria cell is made up of murine. B3. Fungi it is made up of chitin. So 2. And algae it is made of cellulose. So D1. So answer is option A. Then leucoplast are. So these are plastids that are present in the plant cell without pigments. They are leucoplast. And white blood cells, they are called as leukocytes. So don't get confused with leucoplast and leukocytes. So answer is option B. Then the map of 4 genes on the chromosomes are as follows with the map distances given. So between which 2 genes would you expect highest frequency of recombination? So greater the distance between 2 genes, greater the probability that a crossover will occur between them during meiosis. So greater the distance between genes. So A and D. So they are having the greatest distance. So answer is option B. The next one, International Kyoto Protocol of United Nations Framework Convention on the Climate Changes, which came into implementation in 2005, mainly deals with. So it deals with reduction of greenhouse gases. Answer is option C. Then 94, match the following. Fungi, rice blast, option 5. Then bacteria is cholera, protozoa is leishmaniasis, virus is foot and mouth disease. So answer is option A. Then next one, okay, these are the various developmental genes in Drosophila. So Nanos is a formation of posterior structures of the embryo. Yes. Bicod is for the anterior structures of the embryo. 2. Cruppel is gap gene and engrailed is segment polarity gene. So the answer is option B. Then next one. Which of the following mutagenic agents cause thymidine dimer in DNA? So it is UV radiation. Answer is option A. So ultraviolet light, it is absorbed by double bond in the thymine, and this added energy opens up the bond and allow it to react with neighboring bases. If the neighboring base is another thymine, it will result in a covalent bond between the two bases. So mutagenic agent that cause thymine dimer is UV radiation. Then which of the following plant is the source of vinblastine and vincristine, highly valued drugs in cancer chemotherapy? So it is Catharanthus roseus. Answer is option C. Then which of the following sequence correctly represent the phases of a cell cycle? So G0, G1, S, G2 and mitosis. So answer is option B. Then which of the following statements are correct with reference to ruminants? So ruminants, they are herbivores, that is grazing mammals, and their stomach has got 4 compartments, that is rumen, reticulum, omasum and abomasum. And these animals, they chew the cud regurgitated from the rumen, and they acquire their nutrients mostly from plant-based food. So use of microorganism to digest cellulose, that is also correct with regard to ruminants. So that is correct. Then have teeth adapted for mastication, that is also correct. And get their nutrition from digested plant material, no. That is not, yeah, that's also correct. Get their nutrients from, yes, they are plant eaters. So they get their nutrition from digested plant material, that's correct. Then fourth one, eat the feces to replenish nutrients, that they don't do. So answer is option B, that is 1, 2 and 3. Then next one, kinetin is a type of cytokinin that was first isolated from. So it was isolated from herring sperm. Answer is option A. So that's it for this particular paper, paper 2, Life Science, Karnataka SET exam. Thank you for watching my channel. Please do like, share and subscribe. Thank you.